Okay, before we get started on the day's work, I would like to look at the hemoglobin worksheet. I haven't actually graded them, but I have read through quite a few of them, and I found some common mistakes that people need to watch out for in the future. Okay, first of all, the first question was that you were supposed to look at the hemoglobin sequence and then write the sequence of a 20-mer oligonucleotide that would detect the wild type or the mutant in each case. Okay, so there's actually two correct answers to this. One, we're looking at the boldface T in the middle of our oligo, and that's the one we're going to try to detect. It should be in the middle of the oligo because at that position, the correct match is more stable. A mismatch destabilizes the hybrid. So we want to flank that T in this case with 10 nucleotides approximately on either side. So you can either write the strand that you're given, or you can write the complement. Either one will work because we're only given one strand, but both strands are present. So to detect the DNA, you can either give either answer, put the 5' to 3' on there, make the complement if necessary, and reverse the polarity, etc. Okay, so the next question has to do with the RNA, and that's where things went a little bit crazy, where only one of those oligos will detect the RNA, right? The reason is that in the double-stranded DNA from the genome, both strands are present. So it doesn't matter which one you use. You're going to either hybridize the template or the non-template strand. It really doesn't matter, right? For the RNA, it does matter because the non-template strand matches the RNA sequence, and that's the strand that you're given. So you can't use the non-template because it will match, which is to say be equivalent to the RNA, and therefore will not detect it because it can't hybridize, right? What you need is the complement, right, in order to be complementary to the RNA. Okay, so this is the same thing. To detect the wild type, there's two correct answers. Same thing, right? So let's look at the RNA and explain why that is true, right? You don't want to write the non-template sequence because that will be equivalent to the RNA, not complementary to it. So some problems with this part. Some people didn't know what a 20-mer oligo meant. Okay, so 20 refers to the number of nucleotides in the oligonucleotide, 20-mer, and the oligonucleotide is therefore fairly short. So these are single-stranded synthetic DNAs, 20 nucleotides long. Some people complemented a very large sequence, not just the 20 nucleotides, so they didn't know what a 20-mer means. Okay, so that would be a mistake. Okay, or else people wrote oligos that were elsewhere in the gene that weren't anywhere near the nucleotides that we were looking for. Some people started at the ATG. So if you're asked to detect a particular nucleotide, your oligo should be 20 long, approximately, and it should flank it and not elsewhere in the gene. Okay, there were a lot of RNA issues. Okay, some people thought that the DNA oligos wouldn't bind to RNA at all because of the use of set of Ts. That's not true, that RNA-DNA hybrids form perfectly well and are actually more stable than DNA-DNA ones. Some people thought that the mutant but not the wild type would bind to the RNA, and I'm not sure why. I didn't understand why people were writing that, so if somebody would like to explain that to me, I'd appreciate it. But that's not true. All right. Okay, some people thought that both oligos and their complements would bind to both the DNA and the RNA, and that's not true because the fact the RNA is only one strand and will be complementary to only one of those oligos. Okay, so that doesn't make any sense. Some thought that one of the oligos would bind to the DNA and one to the RNA, and that's not true because the DNA has the two strands, so both oligos would bind to the DNA and only one to the RNA. Some people didn't answer the last question at all, making me think that they just didn't know the answer, or some people even said, I don't know the answer, but this is what you said. So that's not the right way to approach that either. So let's look again at what we're thinking about here. Okay, so here's a gene. Always think about the gene. And conventionally, the upper strand is written 5' to 3', although that's not always the case. and we have to consider what happens during transcription. Because so we've got the strand that's being copied is being used by RNA polymerase to make a complement of itself, right? So when transcription happens, RNA polymerase hops on to the template, starts copying in a 5' to 3' direction on the molecule that's being synthesized, and makes a complement to that template strand. Well, if you look at the sequences, the complement of the template strand is, of course, the non-template strand. And therefore, the non-template strand is the one that matches the RNA sequence with the substitution of U's and T's. So if you keep this thought in mind, which is a template, which is a non-template, and which way RNA polymerase is going, and keep track of the sequences, that should dispel further problems with this idea, I hope. All right. So again, get the strand right, whatever. So this is an aside, something you don't have to know, but people didn't want RNA to bind to RNA or RNA to bind to DNA or whatever. In fact, those types of hybrids form perfectly well. But to understand that, I have to remind you or tell you about the other forms of DNA. Okay, we're used to the standard B form of DNA, the Watson and Crick base pairing B form, and we've seen that a gazillion times already. But there are other forms that also occur. Importantly, the A form, which is actually the form that RNA-DNA and RNA-RNA hybrids will take. So there are many cases of RNA binding to itself, forming secondary structures, tRNA being a wonderful example. And when you have that RNA-RNA base pairing, it is in the A form, which has a different configuration from the B. There's also Z, but we won't discuss that today. Okay, but there are some differences. The A form is also right-handed, just like the B form is, but it's squatter and wider than the B form, and it has a lot of sort of chemical sorts of differences. The major difference being what's called the sugar pucker, the actual configuration of the sugar. And if you think about it, that's plausible because RNA has that hydroxyl on it at the 2 position, whereas DNA does not have that. And that's going to drive the difference in the structure between the B and the A form in RNA-DNA hybrids. Okay, so I am no chemist, but I want to just let you know this happens. You'll never hear any more chemistry from me. But the difference is the configuration around that C2 and C3 carbons. Is that in one case, the C2 carbon for the B form is up out of the plane of the ring of the rest of the oxygens. And in the RNA case, it's the C3 that's up. And this is a steric issue because of the 2' hydroxyl on the ribose. So this is from an actual biochemistry book. The steric clash involves the 2' hydroxyl in the B form and therefore driving the A form because of the difference in the sugar pucker. So differences between A and B. A is more compact. This A form has the 3' endo sugar pucker, and the B has the 2'. I should have said this before. The A form bases are not flat along the staircase or helix. They're actually tilted relative to the axis. And there's other physical differences as well. Okay, so it's time to start on cloning, recombinant DNA. And I just want to mention a few reasons why people would even want to do stuff like this, and even what it is. Okay, so we want to mix and match parts of DNA from different sources, different genes, different cells, different organisms, and create new molecules of DNA that do not exist in nature. These will be propagated inside cells and used ultimately to modify the genomes of cells and organisms. They're also particularly important for the use of producing important proteins for therapeutic purposes for sure, but also for studying the proteins themselves. Okay, one of the earliest examples was a case of the human growth hormone, where children with pituitary deficiency are extremely short in stature due to lack of growth hormone. And there were treatments available before recombinant DNA came on the scene, but these involved making extracts from pituitaries of cadavers. So this was extremely awkward and extremely expensive type of procedure. Whereas if you could produce the protein in a cell line, in a industrial kind of setting, without involving pituitaries and cadavers, that's a much better plan. And that has happened, and that has worked really very well for everybody. Right, so that's the idea of producing proteins that are very rare in mammalian cells, produced in very small quantities in a limited number of cells, that they can be produced by recombinant methods and make them available for therapeutic purposes. Of course, we've got human growth hormone, things like erythropoietin, which is used for treatment of anemia, streptokinase for blood clots following heart attack, etc. Many, many applications. Vaccines is also. Okay, so here's your human growth hormone peptide sequence. It's very small. Um, it's kind of big on the big side for hormones, but that's what it looks like when you write it in single letter code. Right. So the idea is you want to clone the human gene. In this case, it will be a cDNA, and use it to produce a lot of protein in some host organism. Could be E. coli, yeast, or mammalian cell lines. The idea is we can get huge quantities of the protein, which makes treatment feasible for the number of people that actually have this disease. So therapeutics for sure, novel gene combinations, transgenic animals, transgenic pests, all of our sequencing projects, etc. So for transgenic mice, for example, mice don't have necessarily the same diseases that people have, so you can create the disease in the mouse by knocking out or mutating the equivalent mouse gene to what is causing the problem in the human. So making mouse models of things like cystic fibrosis and Huntington's disease allow ways of trying to study and understand the disease in ways that you couldn't do just by dealing with humans. Okay, genetically modified plants, of course, we're all familiar with the fact that commercially grown corn and soybean, etc., has been modified to resist insect pests or herbicides. Gene therapy for people is still not here yet. Have been some successes, but we've learned an awful lot about what's gone wrong, that this should be reality in a very short amount of time, we hope. We said that 20 years ago, but it's still true. All right. So the idea, by way of introduction, is that recombinant DNA generally, almost always, starts with constructing E. coli plasmids in E. coli, before we launch into whatever other sorts of things we want to do with that molecule. So the molecules that we deal with now are almost completely artificial, have been changed in many, many ways, with the basic technology of how you recombine or how you put them together and take them apart is almost always done in E. coli. So we need to know the basics of E. coli plasmids and how they work before we get to some of the more complicated things that are needed to accomplish some of these more ambitious kinds of goals. Okay, plasmids and cloning. Plasmids are used to carry genes into E. coli and replicate them so you can get limitless quantities of whatever that DNA is. So when you say you clone a gene or clone a part of a gene, what that means is you're able to replicate it in a cell and make as much of that DNA as you're interested in. Okay, those types of plasmids can be used to produce proteins, but more often they're a starting point for more complicated manipulations. Okay, so we're going to start with the basics, the basics, before we get into any of the more subtle types of manipulations that can go on. Okay, so general cloning, a piece of DNA is attached to an E. coli plasmid and then introduced into an E. coli cell. Right, this is obviously quite simplified. We're not showing any restriction enzyme sites or anything. But the basic idea is you have a circular molecule, a plasmid cloning vector, which is going to be cut with a restriction enzyme. Then you have a DNA fragment that you want to attach to it. You use DNA ligase to attach the DNA fragment to produce a recombinant DNA molecule. So if that fragment is a piece of human DNA or human cDNA, your recombinant DNA is going to consist partly of E. coli-based sequences, but also your human or other organism sequences, which is now part of the same plasmid. Now this gives you some real advantages because once you've made it, you can replicate that molecule for whatever quantities you want. So one important thing about this, once you have a your recombinant molecule in a test tube, that doesn't help you. What you have to do is get it into E. coli in order to replicate it. So that is done by the process of transformation, where the E. coli cells are induced to take up plasmid, or DNA really, from the outside world into the cell, where the plasmid can then replicate and produce many, many copies of that purified plasmid and the purified gene which is attached to it. Okay, so we do this basic manipulation for sequencing and mutagenesis and sometimes for production of proteins, but we're going to need more subtle types of molecules to do the more extreme kinds of manipulations that people do. Okay, so our basic typical plasmid, in fact this is a typical plasmid, but it's got more than the essentials. Okay, what you have to be a plasmid that's going to replicate in E. coli, you certainly need an origin of replication that's going to be recognized by E. coli proteins in order to start copying the molecule for the purpose of propagation. Without an origin of replication, the plasmid will not be able to increase its DNA, it will not synthesize its DNA, and will be lost from the cell. With the replication, when the cell divides, you will produce new copies of the DNA along with the host chromosome as it replicates. You also need an antibiotic resistance gene for a reason that I will explain in a minute. You also need some restriction sites because you have to have a way of opening up the molecule for the purpose of inserting the gene of interest. Okay, now we also have here a promoter, which you don't always need but is often there, and another selectable marker, which can be convenient if you're going to be going into mammalian cells. But the real essentials are the origin and the antibiotic resistance and the multiple cloning site, which contains your restriction sites. All right, so here's a list of the various elements. DNA sequence. An origin, of course, allows the initiation and replication. Antibiotic resistance, multiple cloning site. So those are the essentials, but many will also have promoters for expression in E. coli, promoters for expression in mammalian cells, things like epitope tags, etc., other stuff that we will talk about next time and after that. So we're going to stick with the essentials right now. Okay, origin and replication is needed for propagation of plasmid. So we've got our E. coli, our plasmid, we've transformed it into a cell, and now we want to make many, many copies of that, or let the E. coli make those copies for us. So origin and replication. Okay, and the key here is the origin of replication is where replication starts. But importantly, and of course once replication does start, you've got opening up of the molecule and the creation of replication forks, which make their way around the molecule, ultimately creating making two new molecules where previously you only had one. Now from the standpoint of recombinant DNA, that's important because once you've initiated at an origin, the synthesis goes all the way around the molecule until it's finished. The cell doesn't know what it's replicating. It doesn't care that that's a mammalian gene. It's that which is basically passively replicated along with the rest of the molecule once replication has begun. So the human gene is coming along for the ride based on the fact that this origin is present on the molecule. Okay, so it's passively replicated. The cell doesn't know what it's replicating, and anything else that's attached to it will get replicated along with the rest. So you certainly need an origin. Okay, antibiotic resistance genes, these are used for selection. And there's actually two separate reasons why we need to have these. Okay, one is we need to kill off untransformed cells, and we also need to get rid of those that have lost the plasmids during growth. Okay, so the idea is we add the plasmid to the E. coli cells by transformation. Now, transformation in E. coli is a relatively rare event, and it only occurs in a very, very few of the E. coli cells. So we've got some transformed cells having done our transformation procedure, but the vast majority did not actually acquire the plasmid in that process. So we need to kill off the non-transformed cells in order to ensure that we get a lot of cells which contain our plasmid. The untransformed cells won't contain the plasmid that we want, and therefore we need to get rid of those. So that's the process of selection, where we're going to kill off the untransformed cells. So we use antibiotics to kill the E. coli cells that did not get the plasmid in the transformation process. And the antibiotic resistance gene enables us to do that. It's only the rare cell that takes up the DNA, the plasmid, is able to survive the antibiotic. So our recombinant plasmid is going to contain at least one gene encoding a protein or so that allows the cell to survive the presence of a drug. Now, typically, that will be ampicillin, but there are others that are also used. Okay, the other thing is with selection is that antibiotics will help retain those cells in the culture. Okay, if you just have some transformed cells with the plasmid in it, and you just allow the cells to grow and grow, they will ultimately, eventually, probably lose it. It won't get replicated, and then eventually by dilution, you will lose the plasmid. So it's important to retain the plasmid and select for that process, so that you're sure that all the cells that are in the culture will actually have plasmid. Okay, so I'm circling the transformation part of the process here. You need a selectable marker, which is going to be antibiotic resistance. You need that to introduce the plasmid into the E. coli cells in the first place. So in general about antibiotics, here's a picture of a bacterial cell, and it shows some of the processes that are essential. We've got DNA replication, transcription, translation, and also intact membranes. Okay, so what's mostly used for these things for cells is antibiotics that target cell wall synthesis and antibiotics that target translation. So for cell wall synthesis, bacteria have cell walls that we don't have, and they're different even from plant cells. So there's a whole bunch of antibiotics which inhibit or destroy the ability of the cell wall to be constructed properly. Now, ampicillin is an example of a penicillin-like molecule, and it works by that same mechanism. So in the presence of penicillin, the cell wall synthesis is perturbed because penicillin and ampicillin prevents cross-linking of the cell wall, leading to its weakening, and eventually the cell will die because the cell wall gets so weak. So the molecule, the product of that, is beta-lactamase, which I'll get to in a sec. So cell wall synthesis is a famous and very widely used target for antibiotics in bacteria. The other big one for this purpose is translation. Remember that prokaryotic and eukaryotic ribosomes are different in size from one another, and they, therefore, there are molecules which will target specifically 30S or 50S ribosomes. So the point of this is that prokaryotic and eukaryotic translation machinery is sufficiently different that there are drugs which will target prokaryotic translation specifically and not perturb eukaryotic translation. And so there's a whole bunch of drugs that are available which will either inhibit translation at one level or another. So those are the big ones that are used for recombinant DNA. So in thinking about this, we have to think about the antibiotic and the resistance. We need to have the antibiotic and also a gene which will confer resistance for one reason or another. So for ampicillin, the target process is cell wall biosynthesis. This is an example of a beta-lactam antibiotic, and what it does is it binds proteins that are involved in cell wall cross-linking, and if it binds them, inhibiting their action, the cells will continue to try to grow, but the cell walls will become weak, and the cell will eventually break open and die. So that's what the drug does. Now, the resistance mechanism is ampicillin resistance. It's often, in labs almost always, the result of beta-lactamase enzyme, which is produced by a gene which we would include on our cloning vector. So what the beta-lactamase enzyme does is open up the ampicillin, the beta-lactam ring of the ampicillin, rendering it inactive so that cells that contain this beta-lactamase are able to survive the presence of the drug. So ampicillin has a beta-lactam structure, okay, and so the common resistance gene encodes the beta-lactamase, which renders the drug inactive. So that cells that have that gene in them, in our case on a plasmid, they can survive the presence of the ampicillin, whereas untransformed cells are killed. So that's our drug is the ampicillin, and our beta-lactamase is the resistance mechanism. And that gene is often found on standard E. coli plasmids. Right, so here's ampicillin. It's a beta-lactam antibiotic. And the resistance is because of the beta-lactamase. So the beta-lactam ring is shown, and that the beta-lactamase opens it up and makes the penicillin unable to bind its target in the cell. So this essentially inactivates the drug so that any cell that is able to, that encodes that enzyme is able to resist the drug. Aminoglycosides are often used also, and the target is protein synthesis by ribosome binding. Okay, so I'll show you some aminoglycosides in a second. General mechanisms for resistance for these are not exclusively, but mostly modification by phosphorylation or acetylation and some other things. So we're not destroying it, but simply phosphorylating it is sufficient to make it inactive in its role in binding proteins from the ribosome. Okay, so here's streptomycin. Streptomycin is an aminoglycoside antibiotic active against ribosomes, prokaryotic ribosomes, inhibiting protein synthesis. Right? There are aminoglycoside phosphotransferases, which will act on the streptomycin and simply modify it by the addition of a phosphate group. So that particular form of streptomycin phosphate is unable to bind to the ribosome target, and therefore this enzyme that catalyzes that reaction will confer resistance to streptomycin on any cell that carries it and expresses it. Catamycin is another one. There are several of these, actually. This is Catamycin A. But the same idea is true, that this protein encoded by the gene basically adds a phosphate group to the catamycin, rendering it inactive and unable to inhibit protein synthesis. So the pair of catamycin in the medium and the catamycin inactivating enzyme on the plasmid will confer resistance and allow those cells to survive the drug, whereas most of the cells will not survive. Okay, here's a list of all kinds of different ones. Ampicillin is by far the most commonly used. Inhibits cell wall synthesis. Okay, catamycin is aminoglycoside. It causes mistranslation, etc. Same with several of the others, streptomycin, etc. They may inhibit protein synthesis at different levels by different mechanisms, but that is an important mechanism for distinguishing prokaryotic from eukaryotic translation, and therefore the target of many drugs. Okay, we're back to our plasmid with essential features. We talked about the antibiotic resistance. We talked about the origin and replication. Now we need to talk about some of the other features, namely the polylinker region. I forgot. Okay. How many promoters does a previous molecule have? 1, 2, or 3? Okay, the answer is 3. The molecule has a region which is noted as promoter. It's identified in that way, so a lot of people are going to pick 1. But there are actually 2 other genes on here. One is ampicillin, or the drug resistance, antibiotic resistance, and the other is the other selectable marker whose identity they haven't told us. Now, those are E. coli genes, which have to produce proteins in E. coli, which are then going to produce the enzymes that we need to inactivate the drug. So even the molecule as shown, that the promoter isn't shown, you have to, the fact that it's a gene implies that it has a promoter, and that therefore there are actually at least 3 promoters on that molecule. Okay, so this is a multiple cloning site. The idea is we need to open the vector in order to add the insert to it. And we're going to use restriction enzymes for this purpose for the most part. These are DNA endonucleases that they cut both strands of the DNA. So the multiple cloning site is very convenient because you have a choice of what restriction enzymes that you can use, which can be very helpful when you're trying to clone a piece of DNA. Okay, so here's what it looks like. PUC118 is a standard, very general cloning vector, and this is its polylink or a multiple cloning site. So you can see it has sites where many different enzymes. You have your choice of which one or ones you're going to use for any particular purpose, which could be dependent on the sequence of the intended insert itself or whatever else is available. So this is the simplest possible plasmid. It has an ORI, a drug resistance, and a multiple cloning site. All right. So this one also has a promoter. All right. So expression vectors, this is going to get complicated, and we're going to talk more about that next time, etc. Okay, but the idea is behind this really, and something to think about, is that gene parts are really modular. That if you have a gene, it has a promoter, it has a protein coding region. If you're a eukaryote, it's got introns, it's going to have terminators, etc. Even proteins can be considered modular because they have different domains that can be swapped out or added to things or whatever. So you can't think of a gene as this intact entity where if you mess with it at all, you're in trouble. In fact, you can grab the promoter from a gene and attach it to a totally different gene, and it will work just fine. So we have to get used to the idea that we can swap out pieces of genes or even pieces of proteins to make novel combinations for our own purposes. And promoters are a good example of that. Okay, so here's an RNA, here's a diagram of a promoter from E. coli. And what's shown here is the arrow is the direction of transcription. The blue rectangle is the gene of interest that we've cloned into the multiple cloning site. The upstream purple thing is the promoter. So what's being shown or indicated here is that RNA polymerase is able to bind to the promoter and then direct transcription across the gene of interest in order to produce an RNA, which will then create a protein for us. So that's your basic. If you're going to make a protein, you need to have a promoter so that RNA polymerase can bind to it and do transcription. Without that, you can't produce a protein. You need to have an RNA before you can make a protein. Okay, so if you're going to express a protein in E. coli, I've written eukaryotic protein here, but that's not strictly fair because it could be a prokaryotic or eukaryotic protein, doesn't matter. But the fact of it is, if you're going to express any protein in E. coli, you need to put an E. coli promoter upstream of the gene that's going to be expressed. You can't use a eukaryotic promoter because they don't work in E. coli, and most of the clone genes that you'll normally see are actually cDNAs rather than whole genes. So we need a prokaryotic promoter to produce a protein in E. coli. Okay, so here are some of the popular ones. LAC is very popular. That's a promoter from the LAC operon, and its regulation is very well understood, and that's a very convenient one, partly because it's been around for so long. T7 and T7 LAC promoters are also very popular. Okay, so the promoter part of this is essential for producing a protein from any inserted gene in this particular molecule. It's not essential for replication or for sequencing or anything else, but if you want to produce a protein, that's something you need to provide. Okay, so the promoter can be anything that works well in E. coli. It can be usually a good one, not one that's part of the natural gene necessarily. And sometimes, often, it's important to produce proteins in mammalian cells. So this is taking this thing a step further, where you're going to cut and paste your molecule using the E. coli plasmid in E. coli cells. But if you have in your idea that what you want to do is produce a protein in a mammalian cell, you have to put it together, put the molecule together in E. coli, and then transfer it to the mammalian cell. If that's the case, what you're going to have for your promoter is actually a eukaryotic promoter, not the prokaryotic one. So sometimes you have to plan ahead and choose appropriate vectors for what your intended purpose eventually is going to be.